Thank you so much for joining us here today. A lot of old faces that I see here. Not to say that you're old, just faces that I've known for a while. A lot of new faces, which is fantastic to see. I've been with Bionano for 5 years now, almost exactly 5 years. I joined during an ASHG in San Diego 5 years ago. My responsibility from that day until today has been to determine what you guys need, which sometimes isn't easy. Sometimes it is. Some of you tell me exactly what you need. But that is the most important thing for me. So I will present to you our product roadmap, where we are, where we're going. But I need to hear from you. I need to hear from you either in one-on-one conversations, emails, other means, what it is that you need. Is Bionano delivering on what you need? So a theme for the presentation is what's next. And so where are we today? So we are continuing to invest in all areas of our workflow, from DNA extraction, isolation, all the way up through the bioinformatics and to the answers. And that is a major investment across all of these workflow steps. In the DNA isolation front, with our Bionano Prep SP chemistry that we released earlier this year, we have put our sample prep, our DNA isolation, into the mainstream. It is quick. It provides a lot of utility in terms of samples that get processed, and there's more enhancements and modules coming to that I'll speak to. That tied together with our DLS labeling chemistry make for a very powerful combination to provide the longest molecules that have incredible content in them. We run those labeled molecules through our Sapphire, which currently is able to provide 1.3 terabase pairs per flow cell of total output, 3 flow cells, 3.9 terabase pairs, at the lowest cost in the industry. There's nobody in the genomics industry that can do this at all, and if they do, it's more expensive. And then this year also we introduced our rare variant pipeline, which teases out structural variants at 5% allelic fraction from the output that you can get from a single flow cell. And I'll talk about advancements going on there. So sample prep, a little deep dive into Bionano Prep SP. Those of you that have used it versus using the previous protocols that utilize plug lysis think that it's a complete revolution. I do. Very quick hands-on time. One technician can process 12 samples in a day without harming the technician. We've up to this point, I've validated this on fresh frozen blood, cells. Blood preserved in EDTA was fairly straightforward. Heparin gave us some challenges, but we got that knocked out. That led a way for us to be able to support bone marrow aspirates. So we are in early access there, getting very close to a full launch of that. That, as you'll see with some data, that protocol is working quite well. And in various stages of development, we're working to adopt the SP protocols for animal tissue, solid tumors. There's a lysates provided through the use of the RLT lysis buffer, which is some segment of our leukemia customers are really interested in, so we're adopting that. And we are engaged with partners on automating this protocol. So Mainstream DNA isolation is what I truly think that we have as compared to QIAGEN, which is undoubtedly known for their DNA isolation chemistries. So this is just a comparison for a QIAGEN protocol and kit that Oxford Nanopore recommends on their website for DNA extraction. I know others use other homebrew protocols, but this is the protocol that Oxford Nanopore recommends kind of as a high sample throughput. And on all metrics, whether it's time for the assay, input materials, the read length, we outshine, but especially on the read length. So this is data coming off of a run on a Sapphire instrument. So this is post DNA isolation, obviously, post labeling, run on an instrument, and you see molecule N50s nearing 450 kb. We do see often these 2.6 megabase pair molecules. So if you are a person that's looking for blue rhinos, you will find them here. And then Oxford's protocols kind of operate in this range. So SP provides really clean data, high quality, long molecules, and very well suited to flow through small nanochannels. So for a population of 30 samples, so we use 12878 as a regular human control for whole genome mapping. We in this particular validation studies, we were averaging 56 gigabase pairs per hour. So pretty phenomenal. So if you guys want to take a picture of this slide, tweet, feel free. But this is actually, so the data here is actually BMA sample. So this isn't your clean cells. This is actually BMA sample that came from a partner of ours that we process. So BMA is typically very dirty, and so these were frozen BMAs, and you can see the level of molecule quality that we were able to achieve. This is phenomenal, and we're going to keep on pushing this further. In terms of robustness across sample types, so we've collected a bunch of data from different types of studies that we had done. So this is separate data sets across multiple users, in some cases across multiple sites. And across the board, you see really phenomenal molecule lengths, great data quality, phenomenal throughput. This is run on the G2.3 chip, which we used to call the 3x1300 chip, so on a generation 2 instrument. But as you can see, it's robust to the things that we can throw at it. So the next thing that we are throwing at it, I mentioned, is animal tissue. Preliminary, still work that needs to be done before we get this into early access. But I just wanted to show you some of our data that's coming off recently. So we've been able to really crank up the throughput on animal tissues. What we need to work on is increasing this molecule length. We know where the knobs are to turn. We will do that. It's just a matter of completing that optimization part of our development phase. And we are moving to human tumors soon. We're going to be sourcing that material very soon, and that will help us to assess other quality metrics such as mappability to genomes and SB detection, which we can't do so well with rats because of quality of references. All right, the last thing I'll say about Bionano Prep SP is that as we were developing our protocol, we realized that if we added our stabilizing reagents prior to freezing of the sample, their center of masses, for the most part, increased for those frozen samples. So this is a stabilizer that's offered in the SP kits, but it's offered separately, and it's not very expensive. And so we offer those separate tubes for you to share with your collaborators. So if you have anybody out there that's going to be feeding you samples prior to them freezing those samples, you can send them this little tube of 100 samples worth of stabilizer, and it's going to make your lives a lot better. That's not to say that the data quality without the DNA stabilizer isn't sufficient. I mean, you could do some interesting things with these very long molecules, but if you can get 300 kb molecules with a little drop of a magic potion, why not? Yes.
If we have a choice between ETA and heparin, please use EDTA.
EDTA, absolutely, absolutely, yeah. Thank you for that question. All right, moving on to the other part of the sample prep, our labeling. So with the DLS labeling chemistry, our first enzyme was DLE1, and it's very robust enzyme. We're very happy with it. We have continued to work on creating additional enzymes for various purposes, and I'm happy to say that as of a few weeks ago, we do have 2 candidates that passed feasibility. So we have a DLE2 and a DLE3. We'll share more details about those as we get deeper into the development pipeline. But what I can say is that both allow for a 30% increase in our label density, which in itself is quite nice. You can imagine the benefits that you can get. And we are in a process of assessing bioinformatically the benefits of either one versus DLE1 or in concert together. Yes?
Increase the density, does that sometimes drop the mapping?
No. The instrument is capable of higher density labeling. So the map rates for these feasibility tests for both DLE2, DLE3 with control samples, we're reaching well past 90%. Yeah. All right, so we'll be working more on this and figuring out how to mix and match and combine, and for both structural variant discovery, but then of course benefits in the areas of non-human work, model organisms, where there may be better choices in DLE2, DLE3 with regards to what you're working on. Sample multiplexing, so as we were envisioning more output from a flow cell, we started working on the ability to be able to run more samples in the flow cell, more than one, to help reduce costs and utilize that output in that creative way. And so what I can say is that is an activity that we're funding very aggressively. It's been more challenging than I had imagined to do the chemistry to attach the fluorophore that we really like to the enzymes that we really like, and so we're doing some engineering there, and it's going well. We will have something soon, but we don't have anything just yet. And it's also important to note that although we're helping to support our users that are playing around with targeted CRISPR labeling and methylation detection with some tools and with some know-how, that we have not yet launched this onto our product roadmap, either of these applications. We are close to doing so, and we're soliciting feedback in terms of the requirements and applicability of these applications into your research. So if you're interested, come come see me. All right. Now on to the chip and the instrument. So the Generation 2 instrument that we released earlier this year runs a what we are now calling the G2.3, Generation 2, 3-flow cell chip. There's also G2.2, Generation 2, 2-flow cell chip. And so this is the system that allows you to get 1.3 terabase pairs per flow cell. So there are some advancements here to report. So the output is currently unchanged, but your ability to get to that output is when we launched the instrument, we had spec'd it at 36 hours to get to the 1.3 terabase pairs. We now see routinely for our control samples, but then also for some of our customers' samples, that they're able to achieve the 1.3 terabase pairs in 24 to 28 hours as a result of some enhancements that we've made, both in the SP chemistry, but also in the adaptive loading, the machine learning algorithms that run continuously on your samples within the instrument. So that's faster. And then also the 100x coverage, the 320 gigabase pairs is faster as well as a result of those changes. So we're happy about being able to crank that out. Now, so what about the Generation 1 instrument that many of you have? So I'm happy to report that as of today, we're releasing an ICS, a software release for the instrument controller. We call it ICS 4.9, which is going to open up the total output of a flow cell to 1.3 terabase pairs. So it was 320 gigs. So we're opening up that to 1.3. Again, as a result of the SP improvements, the adaptive loading that we had on the Gen 2 instrument, we ported that over to the Gen 1 instrument. So that's able to really accelerate the DNA through the nanochannels. Now, the Gen 1 instrument isn't fully optimized to take advantage of that loading throughput. So to get that 1.3 terabase pairs takes a bit longer than it does on the Gen 2, but you're able to get the 1.3 terabase. You're able, if you want to do the rare variant pipeline to get the 5% allelic fraction SVs, you're able to get that output from a single flow cell now. And so that's quite enabling. And the kicker is all the chips that you have in inventory are now enabled. Even though they may say 320, they're going to get you 1.3 terabase pairs. So this is something that we were very nervous about, and without the adaptive loading to make sure that DNA goes through those nanochannels, we had a lot of clogging of our DNA. So we worked very hard for that machine learning to do a good job and not overly detrimentally overload the flow cells. So we're able to get that to you with no increase in per sample cost. So very excited about that. Now, so the G2 instrument and consumables, what we're trying to do is figure out how to maximize the most output that we can give you without sacrificing data quality. And so these G2 chips, the current spec, the 1.3 terabase pairs, translates into a throughput of gigabase pairs per hour down here in just shy of 30 gigs per hour. And in many of our control runs, we see that for the most part, we can achieve higher throughputs. We're working on raising these low performers, and we know what the knobs are in our loading software to be able to do that. And my goal much to the chagrin of my development team, is to be able to provide enough output on a flow cell to get you, if you want to get you to be able to do 1% allelic fraction as SVs or larger genomes. And so that is something that we will be pushing on, and early next year we hope to make some announcements there. So we're getting very aggressive. And then also, once we have the sample multiplexing out, you can also imagine being able to do more than one of these 1300 gig runs. So the 5% allelic fraction runs that are kind of the bread and butter in a lot of the leukemia type studies. You'd be able to do multiple of those on a flow cell with sample multiplexing and these throughput increases. All right, so this is where things get interesting as we get all this data together and put them through our pipeline. So we have a list of pipelines, many of which are new or modified. The rare variant pipeline, which we introduced earlier this year, we're continuously making improvements to that. The CNV pipeline, also calls fractional copy gain loss events, as well as aneuploidy. The variant annotation is now coupled with both the rare variant and Denovo pipeline, so it runs right after those pipelines in unison. You don't have to kick that off separately. And then something new that we're announcing today is a targeted set of applications. The first one being the FSHD, brand name of NFocus, and these are targeted for specific diseases or genomic loci, and they're phenomenal. And you'll hear a little bit more about this from me and from additional speakers today and throughout the week. So Denovo, our bread and butter. We're continuously working to improve that. Recently, we've made some improvements into detecting smaller insertions and deletions, but these are phenomenal numbers. I, as an engineer, would like to see this at 100%. Eventually, maybe we'll get there. But this is whole genome sensitivity, which we're happy with. The rare variant pipeline calls all of our structural variants. Our goal has been to achieve at least 90% sensitivity at these 5% allelic fractions, and low false positive rates. We're doing really well with deletions. With insertions, where you see the fall off in performance for larger ranges, it doesn't mean that we're not seeing those insertions. It means that we're classifying them as something else. So we're classifying them as either duplications or translocation events, intrachromosomal. And that's what you can see here. So for all of these here, we're very happy with the performance, and we're continuously looking at areas of underperformance to see how we can do that. And you can imagine higher label density can shift all of these vertical lines to the left for us in terms of sizes of events that we can determine. So this rare variant pipeline has been used on many types of cancers, both by ourselves and by our customers and collaborators. This is just a short overview list. And all of the data is integrated together first at the circus plot view. And this is an actual ovarian tumor sample run through the rare variant pipeline. As you can see, there are many events that you can drill into or filter out against. So we are continuously working to improve the visualization of all these. So we're very soon will be coming out with a whole genome copy number view, kind of what you're used to with the array CGH. We also have gotten a lot of feedback with regards to displaying various information in karyogram, so we're looking at that as well, along with a few other views that help do some population comparisons. So the in-focus analysis. So as happy as we are with our whole genome variant discovery, we realize that if we focus on particular genomic loci or particular disorders, we can get to what some would call clinical grade performance. Now our goal for all of these analyses is 100% sensitivity, specificity, no false positives. And so we chose an approach where we still do whole genome gathering of the data, so it's still DLS labeling across the whole genome, but in our analysis, you can target that analysis and create local maps of the areas that you're interested in. So taking out all of the rest of the noise for the whole genome and just focusing on that region. And that analysis is very fast and it's very accurate. And then you can perform additional analyses later or in parallel. So you can kick off and then focus FSHD analysis, and you can kick off a Denovo analysis or an RVP analysis. And one will come back very quickly, the other one will churn and get you an answer across the whole genome a little bit later. We've also baked in assessments of stable genomic regions and molecule integrity. So to give you a sense of, is the result that you're looking at coming from data that you can trust? And so that's baked into all of these analyses. So the first one that we've selected is FSHD. Very difficult to diagnose disorder, currently being diagnosed by the use of Southern blot, and you'll hear more about that in later presentations. But in order to perform this analysis, one needs to do, at the very least, the haplotyping to figure out which allele you're at, and the measurement of the repeat array, which in itself isn't difficult for us, but it could be for many, given that each repeat is about 3.3 kilobase pairs in length. In addition to that, we also report out whether there are any structural variants or copy number gains of loss or losses proximal to that repeat array, which may help to inform some more difficult cases. And then also reporting out copy number gain and losses for a gene that's on chromosome 18, but is very relevant, especially as one thinks about the possible FSHD2 phenotype. So all of that information is then spit out onto a report, both in PDF format, together with all the map information, the single molecule support for each of these repeat contractions in this case. And you get a PDF report or a JSON, which you can import into your reporting tools together with whatever annotation that you require. So internally, we had run Coriell cell lines with FSHD positive being confirmed 100% of the time, so we did not miss a single one. We also did some reproducibility experiments with different operators performed in triplicate, all the results, all the repeat results, including the haplotype results, were reproducible. And then we went back to our collection of normals. We took 58 normals, presumed normals, not known whether these folks actually had the phenotype. And in 56 out of the cases, we measured a length above the pathogenic length. In the 2 cases, they were just at 10 repeats, so they were marginal cases. So they may not have been phenotypic, but they may have had a contraction. Right, and you'll hear more about this from our collaborators later on today and throughout the week. In terms of the workflow itself, so it starts off with the same Bionano Prep SP, leads on to the DLS labeling, the data collection, and then the data analysis itself takes an hour and a half. So that's very fast. You don't need to do a Denovo assembly first. It does whatever it needs to do within this hour and a half, and often faster. On a compute server, you can cram 16 of these. In our on-demand cloud offering, you can do as many as you wish. Sample to answer in 3 days. You can batch. When you've gotten good with this, you can batch 36 samples in a work week. All right, moving on to the on-demand offering. So we provide a number of ways to perform analysis of our data. There are the compute servers that we sell. There's the custom clusters that we help configure, although that's always kind of its own adventure. Everyone is unique. And so we've also launched an on-demand utilization of the cloud. And really what this is it's baked into Bionano Access. If you choose to run a particular job for a particular sample in the cloud, you can do so just for that job and continue running everything else elsewhere. And the data does not reside in the cloud. It's just used for compute. Data gets sent over in an encrypted way to one of 3 servers. You can choose which server to go on, US, Ireland, or Germany. Once the processing completes, all of the data gets returned in an encrypted way, and that entire instance gets wiped. All the data gets wiped in the cloud. There's nothing that resides there. We think you can that would be beneficial to use it if you're a customer of a service provider. Often you want to do some reanalysis. If you have uneven workloads, if you're working a large genome, so we're able to gather more resources in terms of compute power and memory, where you can, we've done this, 17 gig bread wheat. We've done it. It took a few days, but it resulted in great results. You can do this. And now that you can collect all of that data on a wheat, you can do these all the time. And of course, this Bionano unfocus analysis, since it's very quick, it would not cost a lot to do it on the cloud at all. And if you're doing these targeted analyses, you don't need your own computing infrastructure, which is very amenable to folks that are trying to transition out of karyotyping and FISH, where you don't have large compute infrastructure. If you're doing these types of targeted analyses, you can do those in this cloud environment. All right, so what's next for Genome Informatics? We're going to continue working on improving the SV, CNV performance. We do have a minor release that's coming out this week that improves some edge cases that we had seen. So that's already locked and loaded and it's coming out. But beyond that, we have work ongoing into continuing to improve not only our sensitivity, PPV, but also the turnaround time. Currently, it takes the Denovo pipeline about 20 hours, and the rare variant pipeline takes about 16 to 18 hours. We want to reduce those. We want to combine as much as possible the results for all these pipelines. So a lot of work going on there. You'll be hearing about in the first half of next year. Loss of heterozygosity, again, coming out first half of next year, making a lot of progress there. So very, very happy with work that's ongoing there. I talked about the visualization features, and more of these end-focus analyses are coming. So we're investing very heavily on generating more of these targeted analyses, both in the cancer and constitutional space. So they will be, you will be very happy with the results, I'm sure. All right, so my final slide is getting back to the workflow. So what's next in the workflow? More sample types in SP, more enzymes. We'll get you that sample multiplexing. We will continue working on increasing that yield and that throughput. And we are going to lower the per sample cost. That is without a doubt on our roadmap. We will get cheaper than array CGH. You can mark my words on that. We are going to get you rarer structural variants and more of these end-focus analyses. Thank you.